Hi there, welcome to the Smooth Muscle Simulation. In this simulation, you will learn how smooth muscle is controlled by the nervous system. Using a piece of rat intestine as the model, you will experiment with different drugs. Will you be able to understand why your new friend Julie got sick and explain it to her? But before we start, let's flash back to the restaurant, which will help you understand why you're here today.
It's a pity that Julie can't join us today. I wanted you to meet her. She has been suffering from slow and painful digestion since last weekend when we went to the forest to pick some berries.
Pick your response.
The doctor told her if she ate some Atropa belladonna berries that they may slow down her digestive tract. It has something to do with the smooth muscle in the intestine. But we don't know anything about smooth muscle. Wait a second, aren't you doing your internship at a physiology lab? Why don't you help Julie learn more about smooth muscle?
Pick up a lab coat from the rack. Well done. Now find a glove box and click on it. Hello there, young scientist. I take your presence here as to understand the predicament that your fellow human faces with her digestion system. Go to the myograph workbench. You might wonder why eating some berries can cause stomach ache. To answer that, we will perform some experiments using a rat intestine. Onwards. Click on the rat intestine inside the beaker. Nice. Today, we will focus on the smooth muscle found in the small intestine. But don't worry, we won't use your intestines. We will use an animal model for that, specifically pieces of rat intestine, which contracts in the same way as the human intestine. We need to submerge the rat intestine in physiological solution. During the next tasks, you can hover over pieces of equipment on the workbench to find out what they are called and what they are used for. Click the part of the experimental setup that should hold the physiological solution. Well done. This tank supplies the solution for the water bath. We will immerse the samples in the water bath. But the tank is empty now, we need to add the solution. Pick the correct physiological solution from the two blue-capped bottles and pour it into the tank. Knowing that the muscle is oxygenated, local PCO2 has been set at 40 torr and the pH is set to 7.4, which of the following factors that you could control would be most strongly affect muscle function in the experiment? Exactly. In order to mimic the real physiological conditions, you also need to set the temperature as it is in real life. This means that the sample needs to be heated to 37 degrees Celsius. To achieve this, we will use a thermostat connected to the water bath. Adjust the temperature of the water bath. Place a fresh piece of rat intestine in the water bath using the forceps. Connect the force transducer. Which item is responsible for transforming the mechanical forces into electrical signals? You are correct. The myograph is connected to the piece of intestine by a thread so that when the smooth muscle contracts, this pulling is recorded. That's why it's also known as force transducer. Examine the graph on the screen. Marvelous human. You can see the muscle contraction, because you're using the thyroid solution. It contains different salts, such as phosphates, bicarbonates, magnesium, and calcium. Out of these, calcium is one of the most important, as it plays a crucial role in smooth muscle contraction. Alright, let's get to our missions. Do you want to see what the system looks like in real life? Have a look at the video below. Go to the hollow table. As shown in the image, the increase of calcium ions inside the cells will initiate the smooth muscle contraction. The decrease of it begins the muscle relaxation. It is important to remember the crucial role of calcium in contraction. You humans have 3 types of muscles. Can you guess which of these depicts the muscles that contract involuntarily? Check the theory pages if you need some help. Nice work. Smooth and cardiac muscles contracts involuntary, such as when food goes through your digestive system or when your heart beats. Even though the iris of your eyes and your intestine are both composed of smooth muscle, they behave differently. You may notice that your intestine contracts in a repeated rhythmic pattern, while your iris is not. This is because the intestine is composed of single-unit smooth muscle, and the iris is a multi-unit smooth muscle. You're doing great so far. Do you know how the smooth muscle is controlled by the nervous system? Let's take a closer look. Click on the body to begin the animation. The smooth muscle is controlled by the autonomic system in a double innervated way. You can hover the cursor over the different structures or molecules to see their names. Double innervation means it is being controlled by both parasympathetic nervous system, shown in blue, and sympathetic nervous system, shown in yellow. You may notice one of the differences between both systems. Parasympathetic nervous system preganglionic fibers are long, while the postganglionic ones are short. The opposite is true for the sympathetic nervous system. Click on the parasympathetic nervous system fibers. The neurotransmitter, acetylcholine, is released at the end of the preganglionic fiber and then binds to the nicotinic receptor at the postganglionic fiber. Click on the parasympathetic nicotinic receptor. Remember that you can slide down the lab pad by clicking anywhere on the background. The binding of acetylcholine triggers the sodium channels opening and allows the impulse to travel to the muscle cells. Unlike in the skeletal muscle, there is no specialized connection between the nerve fiber and the smooth muscle cell. The nerve fibers lie along the smooth muscle cells and release the neurotransmitters from swellings in the fiber, called varicosities. In the case of the parasympathetic nervous system, the postganglionic fiber also releases acetylcholine, which binds to M3 muscarinic receptors spread along the sarcolemma in the duodenum muscle. This triggers the release of guanine nucleotide binding proteins and subsequently the influx of calcium ions through the calcium ion channels on the muscle cell surface. In addition, extra calcium ions are released from the sarcoplasmic reticulum inside the cell. This increase in the intracellular calcium ion concentration leads in the increase in the force and rate of contraction. Then, acetylcholinesterase breaks down the acetylcholine released from M3 receptors into acetyl coenzyme A, shown in pink, and choline, shown in purple. Choline will be eventually recovered into the varicosity to prepare more acetylcholine ready for the next stimulus. Now, let's see how the sympathetic nervous system controls the smooth muscle. Just like the parasympathetic nervous system, the preganglionic fiber of the sympathetic nervous system also releases acetylcholine. It also binds to nicotinic receptors in the postganglionic fiber. The binding also triggers the opening of the sodium channels and allows the impulse to continue to the muscle cells. Here, instead of acetylcholine, norepinephrine is released, which binds to alpha and beta receptors. Click on the parasympathetic nervous system fiber to end the animation. Great! You've seen how the autonomic nervous system controls the contraction of the smooth muscle. Please note that both sympathetic and parasympathetic nervous system regulation as well as the molecules involved in the smooth muscle contraction regulation coexist in the same physical space. Now, let's go back to the lab. Welcome back from your expedition into the cells. Let's see if you paid attention to the molecules that are involved in the smooth muscle contraction. Which neurotransmitter is released from the parasympathetic ganglion? Great! Acetylcholine is a neurotransmitter that plays a key role in the peripheral nervous system. In order to regulate the smooth muscle, acetylcholine is released from the synapse of the ganglia and also by the postganglionic fiber in the parasympathetic nervous system, leading to the smooth muscle contraction. Awesome! Now you know how the smooth muscle is regulated. It's time to go back to our main question: why Julie got stomach ache after eating some berries. Go back to the myograph workbench. In today's experiment we are going to use an isolated system of the rat intestine. Look at the comparison below. Parasympathetic nervous system preganglionic fibers are long, while the postganglionic ones are short. They are depicted in blue. On the other hand, the sympathetic nervous system structure is the opposite. These are depicted in yellow. Which nerves are preserved and are functional in our isolated system? You're smart. We won't use the whole circuit shown on the previous animation but only part of it. The long neuron of the sympathetic stem gets snipped after the ganglion, so those receptors are left in the rat and not included in our isolated system. Do you remember that smooth muscle is controlled by two different parts of the autonomic nervous system? These systems use different neurotransmitters that increase or decrease muscle contractions. However, other molecules can also influence this. It's time to learn how they work. Place a fresh piece of rat intestine in the water bath using the forceps. Connect the force transducer. Some people might feel a sudden adrenaline rush if they see this on their phones. Epinephrine, also called adrenaline, can act as a neurotransmitter when it's synthesized in the neurons. It can make the heart beat faster, but it also affects the smooth muscle. How do you think epinephrine will affect the smooth muscle contraction? Read the theory for more info. Interesting thought, human. Let's add epinephrine to the sample to see if your hypothesis is correct. Add epinephrine using the pipette. Check out the graph. You hypothesize that epinephrine will increase the smooth muscle contraction. Is your hypothesis correct? That's it. Instead of increasing the contraction, in smooth muscle, the epinephrine will decrease the contraction. You can see that the waves on the graph are smaller and are created less frequently. This happens because epinephrine binds directly to the alpha and beta receptors, leading to the relaxation of smooth muscle. Place the pipette back and click on the water bath to see the effect of epinephrine on smooth muscle contraction. Behold our isolated system, without sympathetic fibers and only the postganglionic parasympathetic fiber. When adding epinephrine to the isolated system, it's like we aerating the sympathetic nervous system influence. Click on the epinephrine. Epinephrine binds to alpha and beta receptors, and the coupled G protein is released. Click on the alpha or beta receptor. This leads to the cell membrane depolarization due to the release of potassium ions outside the cell, and subsequently to a decrease in contraction. Now let's go back to the lab to continue with the experiments. Hi, welcome back. You just saw how epinephrine affect the smooth muscle contraction. Do you still remember what receptors were involved when adding epinephrine? You got it. Epinephrine binds to alpha and beta receptors, located in the smooth muscle cell membrane. This leads to the opening of the potassium channels and the hyperpolarization of the membrane, which subsequently decreases the intracellular calcium level, thus leading to an inhibition of muscle contraction. Excellent work so far, human. Let's continue with the second neurotransmitter, acetylcholine. Place a fresh piece of rat intestine in the water bath using the forceps. Attach the force transducer. Adjust the temperature of the water bath. As you already know, acetylcholine is a major neurotransmitter in the autonomic nervous system. It's synthesized by the enzyme acetyltransferase in specific neurons, which are also known as IO neurons because of that. Botox can inhibit acetylcholine, thus relaxing the skin and reducing wrinkles. What do you think will happen if we add acetylcholine to the rat intestine? You amaze me, human. Okay, let's check your new hypothesis by adding acetylcholine to the sample. Add acetylcholine into the water bath using the pipette. You hypothesized that acetylcholine will decrease the smooth muscle contraction. Is your hypothesis correct? That's correct. Acetylcholine binds to the nicotinic receptors located in the parasympathetic nervous system postganglionic fiber and also to the muscarinic receptors present in the smooth muscle cells. Therefore, the smooth muscle is doubly stimulated, thus increasing its contraction. Place the pipette back and click the water bath to see the effect of acetylcholine on smooth muscle contraction. Here is our isolated system again. The acetylcholine will bind to the nicotinic receptors in the parasympathetic postganglionic fiber. The binding allows the influx of calcium ions into the postganglionic fiber to transmit an action potential. The postganglionic fiber releases more acetylcholine that binds to the M3 muscarinic receptors on the smooth muscle cells. This triggers an increase in the force and frequency of contraction due to the uncoupling of G proteins and the increase of the intracellular calcium ion concentration. Click on the acetylcholinesterase. Then, as in the normal contraction process, acetylcholine is degraded by the acetylcholinesterase and choline, shown in purple, is recovered in the varicosity. However, since the concentration of acetylcholine is much higher in the medium, acetylcholinesterase can't degrade all of it as fast as in the normal contraction, which leads to an increase in force and frequency of the contraction. Let's go back to the lab again. Hello again, human. After exploring the cells, you may know the answer for this. Which enzyme degrades the acetylcholine once it is released from the receptors? Superb. Acetylcholinesterase helps regulate the smooth muscle contraction by degrading the used acetylcholine into acetyl coenzyme and choline. It's found mainly in the membrane of muscles and chemical synapses of the cholinergic type, like ganglia of autonomic system, and its activity serves to terminate synaptic transmission. Go to the hollow table. Great. You know the parasympathetic and sympathetic nervous system well. What happened to your gut if they're absent? Behold the myenteric plexus. A part of enteric nervous system, the myenteric plexus, releases acetylcholine, which regulates the constant contractions of the gut smooth muscle when there is no influence from the sympathetic and parasympathetic nervous system. What part of the autonomic nervous system is being mimicked when epinephrine is applied to the small intestine? Great. That's it. The sympathetic nervous system controls the smooth muscle by releasing norepinephrine as neurotransmitter, thereby decreasing contractions. Epinephrine is a very similar molecule that also acts as a hormone and neurotransmitter in the body, leading to the same effects as norepinephrine. Good work so far. The doctor told us that your friend Julie ate the nightshade berry. I have extracted the active compound of that berry on the myograph workbench. Let's check the effect of it to the smooth muscle. Go back to the myograph workbench. Attach the force transducer. The active compound of nightshade berry is called atropine. Let's see if this causes Julie's stomach ache. Add atropine into the water bath using the pipette. Check out the graph. As you can see from the graph, atropine decreases the contraction of our isolated system. We also know that atropine is a receptor blocker. Which parts of the autonomic nervous system are being affected now? Yes, atropine is a receptor blocker and, since contraction decreases, it likely only affects the parasympathetic nervous system because we're using an isolated system. If it would affect the sympathetic nervous system as well as the parasympathetic nervous system, we might observe no changes in the contraction signal. Great, we know atropine affects the parasympathetic nervous system. Which receptors are possibly being blocked by atropine? You did it again. Those are the receptors involved in the parasympathetic nervous system, which is responsible with increasing the smooth muscle contraction. However, with the information we have, we can't be sure about which ones are being blocked now. It looks like the atropine slows down the smooth muscle, causing slow digestion that leads to stomach ache. From the previous mission, you learn about epinephrine and acetylcholine. Which of those two can counter the effect of atropine? Add the neurotransmitter that can counter the effect of atropine into the water bath using the pipette. The epinephrine will make the smooth muscle contraction even slower. This could be a dangerous situation. I will reset the experimental system so that it only contains atropine. Okay, after the addition of acetylcholine, there's still no change to the contraction. Therefore, what kind of receptor blocker is the atropine? Remember, we're using an isolated system. Your mind is incredible. Atropine is blocking the muscarinic receptors so no acetylcholine can bind them, no matter if it comes from the nervous system or if it's added externally. This leads to the inhibition of muscle contraction. Place the pipette back and click the water bath to see the effect of atropine on smooth muscle contraction. Here is our isolated system again. When adding atropine, it binds to the M3 muscarinic receptors and, therefore, acetylcholine released from the myenteric plexus can't bind the M3 receptors. Click on the receptor where atropine will bind. This leads to a slightly decreased muscle contraction. Moreover, adding more acetylcholine to the sample has no effect in the muscle contraction, as it still cannot bind to the receptors blocked by atropine. Let's go back to the lab to see if we can find a way to improve Julie's health. Great job so far. The acetylcholine could increase contraction of the smooth muscle, but it's being blocked by atropine. The unbound acetylcholine is then broken down by acetylcholinesterase. To improve the effect of acetylcholine, what should we do? You amaze me, human. Perfect. An acetylcholinesterase inhibitor will prevent acetylcholinesterase from breaking down the acetylcholine, thus enhance the effect acetylcholine. Physostigmine is an acetylcholinesterase inhibitor that often used to counter atropine. Add the solution that increased the effect of acetylcholine into the water bath using the pipette. Nice. As you can see from the graph, adding physostigmine or more acetylcholine can counter the effect of atropine. Let's see how physostigmine would work in an isolated system without atropine to understand its effects on smooth muscle contraction. Place the pipette back and click the water bath to see the effect of physostigmine on smooth muscle contraction. Here is our isolated system again. Click on the physostigmine. Physostigmine will prevent the acetylcholine from being degraded. Physostigmine binds to the acetylcholinesterase, blocking the binding of the acetylcholine released from the myenteric plexus. Since acetylcholine won't be degraded after being released from the muscarinic receptors, adding more acetylcholine to the sample will increase the muscle contraction in both force and frequency even further. Awesome. Let's go back to the lab to wrap up today's session. Physostigmine blocks the catalytic site of acetylcholinesterase, preventing acetylcholine from being degraded. Therefore, what kind of agent does physostigmine act like? Wow, you're becoming a real muscle master. Physostigmine is a cholinergic receptor agonist because it acts like acetylcholine when binding to the acetylcholinesterase. It boosts the acetylcholine action. An agonist is the opposite of antagonist, which is a substance that acts against and blocks an action. You have proven yourself as a good scientist and a good friend. You found that the berries Julie ate slow down the contraction of her intestine and cause the stomach ache. You also found the treatment. Julie is pleased and won't eat recklessly anymore. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.